affect your brain, and you were going to go a little bit crazy. And Al Capone ended up in a, I can't remember what they call those asylums, a syphilitic asylum. He ended up in one of those. Like, why was he in jail? Maybe he should have been in jail. I don't know. I'll have to watch a story about his life. That was just kind of gross but interesting. I like to watch all those historical things like crap that went on way back whenever. So all these terrorist attacks and everything, they're like, oh my God, people are crazy. The whole world is crazy. But if you watch the story of Attila the Hun, and I also watched, they had a show about King Tut on the Channel 57, the Sci-Fi channel or something like that. They had a King Tut program on. And the way these people treat each other is like, they're like terrorists. It's just the same crap. I think it's because men rule the world and have too much testosterone.
Yeah, but
so they're overly aggressive and they kill. Like, and these are brutal. They're killing us.
I know, but we're in the 21st century.
Women just hurt each other emotionally.
I mean, we're in 21st century. Can we stop being like until the
Well, they say, I actually have a movie, guys, the golden year. It's going to come, but not till after we're long dead. It's not going to come for several hundred years yet. That's called the golden year. That's when humans come back around to being enlightened and nice, like the avatars. So the world will be dead.
And where do they get this golden year idea from?
I don't know. I don't know. I forget. Because it's happened before. There's a cyclical nature. It has to do with the movement of the planets, and it's a universal thing.
So the movement of the planets causes us to kill each other. Got it. So I know what to say from that.
Yeah, that's right. Yeah, but humans are not in a good way right now. Alrighty, don't forget who's a eukaryote, who's a prokaryote, who's multicellular, and who's unicellular. Okay, right? Protista, unicellular, eukaryotes. Unicellular eukaryotes, protista. Fungi can be unicellular eukaryotes, yeast, or multicellular eukaryotes, molds and mushrooms and other fungi. And then you got Monera, or bacteria, and those are unicellular prokaryotes. And animals are multicellular eukaryotes. Did I miss anybody? Plants are multicellular eukaryotes. And don't forget about autotrophs and heterotrophs. What's an autotroph do? And what's a heterotroph do? Right? The photosynthetic organisms, you know which ones are photosynthetic, your algae, and different types of algae. Alrighty, let's look at some metabolism. So did you learn your metabolism? Right? Catabolism is what?
When you break the big molecule to make small.
Right, you take a big molecule and you break it, you make it into smaller molecules, and when you break those bonds in the big molecule to get smaller molecules, those bonds contain electrons, and hydrogens follow electrons, and electrons represent energy. And hydrogens can be used to generate energy to make ATP. So catabolism is extracting energy from the bonds in your food. You break the bonds in your food and release the energy that's in the food. And then you store that energy as ATP. And then what are you going to use the ATP for?
For anabolism.
For anabolism. So you're going to make big molecules from small molecules. What kind of molecules do you want to make?
Proteins.
Proteins and DNA. I make proteins and DNA. You also want to make RNA because you need RNA to make proteins. So, alrighty. And in catabolism, if the organism can use oxygen to perform aerobic cellular respiration, how much ATP does it get? How many?
Use oxygen, you said?
Yeah, if it uses
38.
oxygen, 38. It's 38 per what?
Molecule.
Per molecule of
Glucose.
Glucose, good, get 38 per molecule of glucose. Okay, who's the final electron receiver when oxygen's available?
Oxygen.
Oxygen, right, O2. Okey-dokey, where do you get your 38 ATP from? What reactions?
Glycolysis, Krebs cycle,
Electron
Electron
transport chain.
transport
So you
chain.
get 2 from the glycolysis reaction, and you get 36 more from doing Krebs cycle and oxidative phosphorylation. Those 2 are the reactions that require oxygen. So with oxygen, you get 36 more ATP than you would get without oxygen. We're talking about bacteria with the 38, and even eukaryotes get 38, but remember they have to pay 2 back to enter the mitochondria to do their Krebs cycle and oxidative phosphorylation, so their end result is 36, not 38. But we're going with 38 for bacteria. What if you're anaerobic and you function without oxygen? What's your total ATP yield?
2.
2. And where did they come from?
Glycolysis.
Glycolysis, right. What happens what's the second step after glycolysis without oxygen? What do you gotta do?
Fermentation.
And fermentation means what?
That means that you reduce the pyruvate.
Good. That means that you reduce the pyruvate, exactly. The electrons and hydrogens that otherwise you would use to make more ATP, you're gonna have to tie them up because you don't have oxygen to attract them away. So instead, they're going to get dumped onto the pyruvate, and they're going to ferment the pyruvate by reducing it, which means they add electrons, so they become increasingly negative. So reduction means you gain a negative charge from gaining the electrons. And the reduced product, when you reduce pyruvate, what do you get?
Alcohol plus CO2 or lactic acid.
Good, good girl. She knows her stuff. Alrighty. What if this little microorganism, like E. coli perhaps, or other members of family Enterobacteriaceae, have enzymes that enable them to run aerobic cellular respiration, so if they're cut off from oxygen, like being stuck down in the bottom of the auger, they can also run anaerobic cellular respiration. What kind of metabolism is that called? When they can do both. Facultative anaerobes, good. Alrighty. Sometimes you'll get a little scenario. You grew this on your Petri plate, and then you made a slide of it. What do you see? Or you grew this on your Petri plate. What does it look like?
Mixture,
Like
the mixed culture and food culture.
yeah, and also individual organisms. What happens when you stain an organism? What happens when you stain an organism? Yeah, you increase the contrast, and what's going to happen? You're going to override its natural color. So let's say we pick on Katie because she's got on this beautiful light coral shirt today, and we dip her in some methylene blue. Well, she's probably not going to look coral anymore. She's going to look more like Yosef with the dark blue sweatshirt. So when you take bacteria off the Petri plate and put them in a slide and stain them, they're going to pick up the color of the stain. They're not going to stay their natural color. So you're going to override their natural color. So if I have Micrococcus luteus, they're yellow on the Petri plate. I take some of them off, make a slide out of them, stain them blue. Well, guess what? They're not going to be yellow. They're going to be blue from the stain. Alrighty. And forget your structures. There's structures of bacteria, there's structures for protista, there's structures for viruses. Remember protista, we classified them by usually the structure they have for movement. Some protista had flagella, and some protista had pseudopods or pseudopodia, and some protista had cilia. Bacteria, some of them had flagella, but bacteria never had cilia. Some bacteria have fimbria, some bacteria have a pilus and a plasmid inside, and some bacteria have a capsule, some bacteria have inclusions, all bacteria have a nucleoid, that's the bacterial chromosome, and bacteria have free ribosomes. If we look at viruses, some viruses have a capsid, well, all viruses have a capsid. So don't confuse capsid with capsule. some bacteria have a capsule. All viruses have a capsule. Some viruses have an envelope, and some envelopes have protein spikes, or glycoproteins, your book calls them. And some viruses also have an extra inner core covering their nucleic acid, like an inner capsid almost. And some viruses have extra enzymes inside. They have some special features. Alrighty, let's talk about how are you going to kill your microbes? If I buy my meat at the grocery store, bought a big thing of hamburger meat, and I have it in my shopping cart, and then I run into one of my girlfriends from the gym, and we start gabbing for half an hour, and then I get in the checkout line, and it's taking forever, and then I finally get my groceries bagged up, and I go out to my car and put them in the trunk, and then my damn car won't start. Then I have to wait for AAA to come and jump and start me or whatever, and then finally I start driving home and I hit a red light, and I'm stuck at the friggin' red light because some old fart wouldn't turn. And then finally by the time I get home, I take my meat out of the trunk and I throw it in the freezer. Did I kill the bacteria that grew in all that time?
No.
No. What's going to happen when I take that meat out and I let it thaw on my counter? Yeah, those bacteria are going to like get unthawed there, and they're going to start growing and eating meat and excreting waste into it, multiplying in number, all that stuff. Good. So freezing is not going to kill them. Also, how do we store viruses? We freeze them. Remember? And the guy's sitting in the tree catching mosquitoes, and then he puts them in nitrogen tank to freeze them, and they take them back to the lab.